here. I'm gonna try and get us started off right here. Sorry, I got a little screen. I'm gonna get things. All right. Hopefully this works out well. So before I even introduce myself, which I guess I did in the chat already, I wanted to kind of frame us in the mind and the place of freshwater mussels. So I'm gonna play this little video. I think it should work. Does not look like it's going to work. Hold on, let me try this real quick again. Let me see if I can start the video and then start sharing. Maybe that'll work better.
Technical challenges.
Oh, I know. I'm much more of a running around the room kind of presenter than a sitting in the chair presenter. But interesting. I guess I already broke something. So I can't get that to start. Good start, but we will give it a round two start here with the title slide that is much more static and should be up right now. There we go. So that slide, that static slide, looking pretty good to you, Alexa?
Yes, that looks great.
All right,
nice and clear.
Thanks. Yeah, so I'm Teal Waterstrat. My day job is working for the U.S. Fish and Wildlife Service, and taking off that hat for the evening here to talk to you about freshwater mussels. But that is something the Fish and Wildlife Service offices in the West Coast are getting much more engaged in right now. I wanted to give a big thanks to the Grays Harbor Stream team and also the Pacific Northwest Native Freshwater Mussel Work Group, one of those long named organizations, but a great resource for those of you interested in freshwater mussels. So on the title slide, these static picture are of some of our Chellis freshwater mussels. Let's see if this is their valve in there. So I just wanted to give you a quick introduction to those guys and then an introduction to the talk. So tonight I'd kind of just like to quickly tell you what freshwater mussels are as a group and in the Chellis. A little bit of information about why the Chellis is such a really interesting spot for freshwater mussels in Washington State. Let's see, what else I was going to tell you. I was going to tell you why they're important, introduce all the species of the Chellis Basin to you, give you a quick update. There's been a lot happening in the past year and a half in the Chellis Basin for freshwater mussels. So I kind of want to introduce you to some of the new work that's going on, because it is kind of the cutting edge of freshwater mussels on the West Coast right now. And also tell you how you can help freshwater mussels. Kind of the excitement Alexa was talking about after last year's presentation led to a couple new observations of new freshwater mussel populations that we didn't even know about. So there's really an easy and direct way you can connect freshwater mussels and watershed health and all the mission of the Grays Harbor Stream team together, just by simply knowing what freshwater mussels are and letting others know that they're there. So I'm going to quickly try and explain the exciting world of freshwater mussels to you. And those of you on the chat, have any of you seen this presentation before? I don't know if you can make the fun thumbs up sign or say yes or no. No worries either way. Try and keep it entertaining. I know we've all had a long day of Zoom meetings already, probably. So I'll try to make this pretty fun. But
Laura says no.
Nope. Great. This is all new then. I won't have to bore
Kurt
you too much.
says yes, and Martin says no. So
all
we
right.
have a mixed audience.
Mixed audience. And I should say before I get started, about 10 or 15 years ago when I started really engaging in freshwater mussels, there wasn't very much knowledge or expertise in my profession and conservation biologist profession on the West Coast. And that has radically changed in the past 10 years, not because of anything I did, but because of a lot of partnerships and a lot of really dedicated individuals just bringing information like this to the group. So if some of you are those people out there and you see me going astray of the latest and greatest science and information about freshwater mussels, feel free to interject, because I'm becoming one of the old timers rapidly that used to be very knowledgeable, but now I'm quickly becoming one of the less experienced and less knowledgeable people out there, just because of the pace of stuff. So freshwater mussels, what are they? They're bivalves, mollusks, bivalves, just like the marine mussels and clams, except they have a completely different life history than any of your marine bivalves. They're really long-lived. Some of the species in Chellis I've personally aged out at over 80 years. So a lot of these individuals are sitting in place at the bottom of the river for nearly a century. And we'll talk about why that's a really cool aspect of their life history a little later in the talk. Like all the other bivalves, or most of the other bivalves out there, they're filter feeders. So they are always pumping water in and out, looking for the right little bits of nutrients flowing down the rivers and in the ponds. And also North America is actually the world's hotspot of freshwater mussel biodiversity. A lot of the times, we see these amazing films and documentaries about the biodiversity of Africa or tropical South America, or these huge other biodiverse places, coral reefs, but North America is the hotspot for freshwater mussel biodiversity. And this last bullet is a bullet from a while ago, where it says they have a great life. What are they? And what are they doing? They're having a great life in the Chehalis, but unfortunately, later on, we'll talk about some of the recent declines we've seen, not just in the Chehalis, but in the numerous rivers in the West. So I still think it's a great place for freshwater mussels, but things are a little less tranquil than they were 10 years ago. So this slide right here kind of introduces you to the roughly 850 species of freshwater mussels across the world. You can see my cursor, is that correct, Alexa? That's probably not the fanciest, kind of circling in the top.
Yes, I can see it.
Okay,
yeah.
Great, thank you.
Mm-hmm.
Anyway, yeah, so North America has nearly 300 of the 850 species of freshwater mussels in the world, but only 2% of those 300 or so species are in the west of the Rocky Mountains. So we only have like 6 species over here. This Appalachian, Mississippi area has the majority of those 300 and some species. We've lost a good chunk of those already, and there's a lot of freshwater mussels that are in trouble right now. So it's a very at-risk group of organisms or family of organisms. And feel free to interrupt me at any time. So briefly, I compared our freshwater mussels to marine mussels and oysters and other bivalves we're more familiar with. And one of the most exciting ways that freshwater mussels deviate from those other life histories or other species is in their kind of reproductive life history. This diagram, which takes a little bit to study, I wanted to walk through it with you, kind of shows their reproductive life history. So down here, number 1, they're bivalves, they're broadcast spawners. The male releases sperm into the water, and hopefully there's a female downstream there that will receive that and fertilize her eggs. And that's pretty common for a lot of marine organisms as well, bivalves. And this is kind of where things get very interesting for freshwater mussels, is instead of releasing those fertilized eggs out into the water to meet their own fate, freshwater mussels have a much different approach where they actually use a fish, most of the time a fish. There's a few exceptions out there in the world, but most of the time they use a specific fish or fish species or set of fish species as kind of a host. And what these little baby mussels called Glaucidia are doing they're latching onto the fish, and a lot of them are actually sucking nutrients out of the fish. Like they'll attach directly to the gills in this case, and they'll be sucking nutrients out of the fish and helping them grow into juvenile mussels. And in addition to this kind of like parasitic role these juveniles have, the fish actually move the mussels up and downstream from where the parent is found. If you think about that, how would mussels get upstream? How would they work their way into our coastal foothills and into the Cascades and into the Olympic Mountains? And they do that by hitching ride on fish. So these little mussels are kind of like vampires riding on a fish school bus up our streams and rivers. So it's pretty fascinating that these mussels have evolved to use fish as both the means of moving themselves throughout watersheds and between watersheds, and also as a kind of a nursery for their young. And once the Glochidia develop enough on the fish, they drop off, presumably in good fish habitat as well, which is kind of interesting to think about. They drop off the fish and start to grow as juvenile mussels in the substrate. As they get older, they'll kind of come out of the substrate and start filter feeding a little more aggressively. Want to see if anybody had any questions or comments on that really quickly?
Yeah, question, Teal. What timeline is that they're actually spawning? And I'm trying to figure out what species they would be attaching to.
Yeah, sure. So those are still somewhat mysterious. We are starting to get more information on that. And it seems like they're spawning pre-spring quite a bit, or in the springtime, but it seems very variable on water temperature and presumably some in cue, the fish itself or other environmental cue that lets them know there's host fish present. The fish itself is fairly variable between species. Some mussel species have one host fish, one species of fish that they can use as hosts. Others are much more general. The pearl shells, which I think we saw a picture of in the title slide, are very much salmonid specialists. So they can use salmon, trout, what was the other salmon-like species, char, they can use as well. But they're kind of stuck in that salmonid group. One of the things we're starting to realize in the West Coast more is we'll find those Glochidia on numerous fish species, but kind of like kids usually are, they're pretty indiscriminate. They don't really know what's going on. So sometimes they'll attach to a fish that will not serve as a host fish, and they will not grow. Essentially, they'll attach to a fish, but it won't be the right fish, and they're unable to complete into successful juveniles. So they can be pretty specific. I can mention a couple of the other host fish guilds, but that's a body of research that's actually just kind of growing right now in the west. Sorry, not the west. So yeah, great question. I wish I had all the answers, but that's another reason these species are kind of fun to work with, because there's great questions that haven't been answered yet that we really need to know. So I want to give you kind of another snapshot of that. There's a really cool video, and I don't know if there's a way to move. I'll send that to Alexa, and maybe, she can put that in the chat in a bit. But it's a really fascinating video, but I didn't want to force you guys to watch a video. Well, you were watching a video of me already. That just seemed like a little too many videos overlaying on yourselves. We live such a screen time dominated life right now. So I'm gonna spare you that, but I will send that to Alexa afterwards. And it's a really short video, but it's amazing. It does a lot more than I can ever do with words to show how unique that evolutionary process is and how amazing the juvenile mussels are. But this slide still has some interesting pictures I want to share with you. This is I believe a Chellis mussel, a freshwater mussel from the East Coast. And a lot of mussels actually during the time that they're have fertilized eggs will grow this fish lure on their mantle. So this is kind of almost like birds changing their plumage seasonally. Female mussels grow these fish lures, and they look very much like their prey items of their host fish, believe it or not. And they will wiggle and waggle these lures around. Some of them look like crayfish. Other of them look like crazy disco parties, kind of the fishing lure variety of things to attract specific host fish. And that's one mechanism for these mussels to get that specific host fish to get close enough to them to release their conglutinate or package of baby mussels onto the fish. So it's really fascinating to start looking at these and seeing how well organisms without any visual senses can mimic a real fish. Just think about over the eons trying to make a fish-like lure without ever seeing an actual fish. Up in the top left here, this is one of our pearl shells, and this is its conglutinate or pack of baby mussels, essentially. To me, this looks a little bit like a bit of a worm, maybe something a fish might come over and hit and allow those babies close proximity to the fish and attachment. And then this picture just shows a little zoom in of these Glaucidia squeezing onto the gills of a fish, in this case a bass. But this is kind of the vampire mode when they're like clamping on like little Pac-Mans to different parts of the fish and starting to use the host fish nutrients. I did want to mention a paper just came out looking at Atlantic salmon and a sister species of pearl shell mussels in the Chehalis that found that this parasite load of juvenile mussels doesn't really affect the health of the salmon. It's kind of a short period, and they shed those juveniles, and seemingly the gill tissue recovers very quickly. So I know there's a lot of perception in the fish health and fish propagation community that these may be parasites and detrimental to juvenile fish salmonid growth, especially. But that doesn't appear to be the case within this genus. But that's another really fascinating aspect I think could be explored a little bit more, is how much this parasitic stage affects juvenile salmon. But we'll talk about a lot of reasons these freshwater mussels are very important for salmon health a little bit further on. So in this little diagram, this is probably very familiar to you because it's Gray's Harbor Stream Team, very familiar with the Chehalis, but the Chehalis itself is as you probably know, a very biodiverse river. It's got a couple species in there, the mud minnows, a number of amphibians, a number of freshwater fish and mussels that are pretty absent in a lot of adjacent drainages. And I think, there was the giant glacial scour in the Ice Age that came down just about to the Chehalis. And then, of course, there was a Missoula flood that wiped out just about everything else but the Chehalis again. And I think, the reason this basin, and again, this is pretty speculative, but kind of makes sense, is the Chehalis has dodged a lot of bullets. It's dodged an Ice Age, it's dodged major floods. And for that reason, it seems like we have more freshwater mussel species in the Chehalis than most other rivers west of the Cascades. So it holds, the Chehalis is kind of unique in holding all three genera of freshwater mussels found in Washington State. And I think the next slide will take us into those three genera. Yep, all right. So in the Chehalis, we have Margatifera falcata, and we're gonna learn a little bit about each of these species. Can you guys still see me or just my screen? Or is it variable?
I can see you and the presentation slides.
Okay, sorry. So yeah, we'll learn a little about this fellow, the Western pearl shell. We'll learn about the ridged mussel, which has this nice, let's see if I can get it. Ridge, I guess that's not showing up too well. And then we'll learn a little bit about the third one, the floaters of the anodonta. There they go. So those are our three, and it's really nice. That slide that showed we had 300 and some species of mussels in North America. One of the great things about being out here is, despite a lack of biodiversity, we can all become experts on all the freshwater mussels in Washington State, California, Oregon, British Columbia, Alaska, Idaho, a chunk of Montana, most of Nevada. We can learn all of that tonight in about 15 minutes or so. So you can kind of be West Coast specialists by the end of this presentation. And I think Alexa has a training certificate that says that at the end of the meeting, right? You all get credit for that. It's an official Teal Waterstrat training certificate, so carries a lot of weight out there in the world. First off, the Western pearl shell. So we're going to present these in kind of fast to slow, or thick to thin is how I like to think about it. So we're going to start off with the fast and thick mussels. So that means they're living in faster waters, and because they're in faster waters, they get banged all the time. So these mussel shells are thick and hard and resilient. So they're in those fast waters. The Chehalis, when it's ripping down in November or another flood season, they're enduring that mainstream high flow, kind of high velocity river habitat. There are a lot of the main stem rivers, a lot of the tributaries have these right in that kind of coarse habitat that you can see down there. They're fairly large and they're pretty dark. Hopefully this is coming through on your screens. This is a bed of freshwater mussels near Elma and the Chehalis. And you can just see how packed in these mussels are. There's a rock here and a rock here, but most of what you're seeing on the bottom of the river right here are freshwater mussels. And they were just so many mussels down there. It was almost unbelievable in the main stem. The video I was trying to show you was the perspective of one of these mussels at the bottom of the river, just kind of watching the river float by with the mussel down there. This is also the longest lived species of freshwater mussels. These are the ones that live or can live to 100 years. We've definitely found them up to 80 years, but that wasn't very comprehensive with the search. So I bet there are some 100-year-old mussels out there in the Chehalis somewhere.
Hey, Teal, just a quick question.
Yeah.
How do you know how old they are?
That's a great question. And the coarsest way to determine that is much like a tree. The shells put down annual growth or almost annual growth every year. So you can do a cross section of a mussel shell and kind of smooth it out, stain it, and put it on a slide, and then count rings. And it looks a lot like a kind of like tree rings, essentially. There's been some really, so I'm getting off my talk now, but there's been some really cool studies that have looked at upland trees, pearl shell mussels, geoducks, and rockfish out in the marine areas. So they've kind of gone from the uplands to the rivers and then out into the coastal areas. And they've been trying to reproduce like chronologies of past climates. These shells last a long time. So they're looking over 100-year periods at like the growth rings in trees, mussels, geoducks, and rockfish otoliths, and kind of trying to piece together upland, riverine, and marine kind of climatic and conditional history using these mussels. So very cool stuff you can do with aging mussels. That method is not perfect. Some years mussels don't grow. As we said, these mussels get banged up in the river. So this is a pretty good one, but you can see a lot of the rings right here are probably already been banged off or eroded away off the mussel. So it's a good estimate, but it can be hard to get the exact true age of a mussel. All right, next up on the Chehalis inventory is our Western ridge mussel. And it's a lot more obvious here in this picture, which is great, than the shell I was trying to hold up. But this mussel has a nice ridge down the side of it. There's other pearl shells out there. There's other floaters out there. There is only one ridge mussel in the world, and that it's, for whatever reason, it's west of the Rockies in North America. So the only place in the world these guys live is west of the Rockies and Sierras. So it's a Western species. It's the only representative genus. So it's our kind of precious and sacred like species. Like this is the only place in the world they occur. They do range from California into British Columbia, but nowhere else in the world. Couldn't tell if somebody was asking a question there, but and
No,
these
it's just me.
species, huh? Sorry.
No, it was just me.
Okay.
I was
And these
just messaging,
species are
sorry.
kind of our intermediate species. These are the hardest mussels to find. They love those soft sediment edges on the side of rivers, on the side of lakes, and you'll barely see any of them. This picture here is from that same great place near Elma, but we've washed away the sediment to find the ridge mussel. You can see all the fine, loose kind of sediment that was around it. It was kind of tucked down in there. These are so hard to find, but very cool to find. So they're kind of not in the main flow the way the pearl shells are, but they're kind of on the edges or in slower parts of the rivers, and can be found along the edges of large lakes as well. And last up, we have our floater. And it's called a floater or anodonta. Anodonta is probably any orthodontists out there or dentists out there know that donta is tooth and A is without. And I forgot to show you this earlier, but anodontas don't have a tooth inside of their shell. All the other ones, it's really hard to see. There's a bit of a tooth right there. Sorry, it's very difficult to see, but it doesn't have a tooth on the other side of its shell either. And these are the thin shelled. It's a very thin shell. still water breeders. So these are mostly found in lakes where there's very little flow. There's a lot of soft sediment. So they don't need to put that much effort into building their shell, and perhaps even a heavy shell is detrimental because they might not be able to stay on top of some of the sediment they live in. And I think probably most of you have walked into a really pretty looking pond or lake, the very edge, and taken that first step, and then all of a sudden you're up to your waist in like slimy, goopy mud. That kind of false bottom that always tricks me. But you'll find a lot of the floaters in that habitat, that really loose sediment. And the reason they're nicknamed floaters, or at least one of the reasons we think they're nicknamed floaters, is their light shell lends them, when they die, to float up above when they're kind of decaying. So, which behind a lot of the big river dams, when they do the drawdowns and then fill the dams back up, a lot of these mussels die behind the drawdowns. And then as the water comes back, they'll just be these kind of little pools of floaters floating around in the back eddies of those reservoirs. There's another great picture of the floater from, I think that one's in Lake Quinault, yeah. So I'm gonna stop sharing for just a sec, because I think that'll make my face bigger. Is that true? All right, so I think we ran through the 3 species real quick. I did forget to mention host fish. So I wanted to mention pearl shells, and they're the salmonid specialists. The ridge mussels here, they are, we actually know very little about these, but it seems like freshwater sculpin are a really important host species, but that's just something we're trying to figure out right now. And then the floaters, and they seem to be pretty much generalists. You'll see them on a lot of different fish, mostly stillwater fish, but they seem pretty generalist in nature. There's a really fun demonstration I like to do, or at least I think it's fun. Maybe everybody else hates it. But one of the things we wanted to do today was show you all 3 genera of freshwater mussels in the Chehalis. And you can actually pretty much tell most of them apart with your eyes closed. And one of the easiest ways to do that is if you take the floater shell here and the pearl shell. These pearl shells, as we know, are the fast and thick and tough mussels. And the floaters are the thin, kind of less rough and tumble mussels. And you can realize that just, let's see if I can do this, just by squeezing. And the floaters just kind of crumble apart. And you can have a kid, not that I'm any sort of Superman, but you can have a kid jump up and down on some of these pearl shell shells, and they're just so thick and dense. I don't know if you can maybe hear the difference. They're impossible to break. Pearl shells also have that nice little tooth inside. And of course, in the middle there is the ridge mussel, and you can kind of feel that that ridge on it. So now you can see them with your eyes, with your ecological knowledge of where they live, and also just by touch. I think you'll be able to tell them apart. I should pause to mention some exciting state regulations. So you can collect all shells that you want to. That's as you want to collect them, look at them, learn from them. We're not supposed to move live freshwater mussels without some sort of permit. So there's no fishing, no recreational or commercial fishing season for them or anything like that. But shells, they're great. They document species presence. But do spend time to look at mussels because they can be kind of fascinating, the live ones in the beds. All right, I'm gonna go try to skillfully navigate back to my presentation. I think I screwed it up already. Let's see. I'm gonna navigate back to my presentation here without, let's see, share screen first. Oop, come on, magic button. There we go. Does anybody have any questions about freshwater mussels species in the Chehalis? So that's it. You got all 3. You guys in the Chehalis there have everything west of the Rockies.
So question on your Western Pearl or in the Chellis Basin, do you have like upper and lower limits of distribution? And the other question is, are there any tolerance to marine environments, tidal influences?
Yeah, that's a great question. So we do have pearl shells as far down as the Highway 107 Montesano Bridge. They may be further down, but I've never seen them down there. So they do have some resilience to being out of the water for some time. They can handle a bit of that. They definitely seem more abundant. I would say from the Satsop upriver, they definitely seem to be more abundant. Once you start hitting that tidal influence, whether that's marine tolerance or brackish water tolerance or tidal influence, or it's just getting a little too muddy down there for this particular species, they do seem to taper off after that. Upper limits to their elevation, I think it's more limited by fish distribution and fish passage than any other aspect of the system. So they're definitely into the Cascades. I mean, at some point, the rivers are too steep and they just get scoured out. But I used to know the elevational record for freshwater mussels, but they get pretty high up there and definitely over the Cascades or around the Cascades. So they can be pretty high up in the basin. Trying to think a good example of that, but they do get pretty high in the basin. And I forget, there was a second part to that question too, I think.
No, you answered it.
Okay, thanks. Trying to think if I was going to say anything else, but I can't remember if I was going to or not. So we know the mussels now. We know a little bit about what's in the Chellis, hopefully a little tiny bit about their life history and what they are. But why they're important to us, why they're important to the Chellis and the entire freshwater ecosystem is something it's a really cool story. So we talked about them filter feed, they're filter feeders. So they're cleaning the water for us. They're filtering out particulates in the water 24/7. Just kind of like, I think there's the liver of the river is one of those kind of nicknames they've been given from time to time, because they're pumping and cleaning water all the time. They're also concentrating nutrients. So not everything they siphon in and out, they keep. They also reject a lot of that food, and they will actually kind of deposit that. It's called pseudo feces because it's not coming out the way it comes out of us, but it's coming back out of their mouth essentially. And those pseudo feces in a lot of our cold, clean, clear streams are actually more like Cliff Bars for other life organisms, because all of a sudden there's this little packet of nutrients the bacteria can start growing on, then invertebrates can start grazing on, and then the fish can start feeding on the invertebrates. And there's been some really neat studies with lampreys as well, showing that like lampreys are actually growing a little bit faster and healthier is the right word, but they're growing more quickly in areas of freshwater mussels beds than outside of it. So there's some really cool like aspects to freshwater mussels and their kind of foundational piece in the freshwater ecosystems. I kind of blew it. I talked about how they can be system sentinels a little bit. We talked about that study where they were looking at trees and freshwater mussels and geoducks and rockfish and how they keep their records of kind of like the current history in their shell. So if we had a midden of these, the beds sometimes are built on old beds, kind of like a oyster reef, not in the attachment sense, but just layering of mussels. We can look back in time to see kind of a record of the river in some ways. Mussels, as you said, are filter feeders, so they bioaccumulate things. So we can actually look at mussel tissue and the shells in some cases and see if there's been like a catastrophic spill in the past. They'll still have information about past events in their tissues as well, or can tell us if there's some mineral or other element in the water or molecule in the water that's maybe dangerous to us. And they can tell us a lot about fish presence too in a lot of our modified landscape. Sometimes we'll find freshwater mussels above where we find fish. And that's because, these are long-lived organisms. Perhaps 50 years ago, 100 years ago, a dam went up or something like that, and fish or a culvert was placed, or a road was gone over, some other change happened in the environment. We can look back and see that, hey, this site hasn't had juvenile freshwater mussels in about 50 years. What happened 50 years ago? what changed the ecosystem. And we can go back and look and see like, what we can change to bring both the fish back so we can have mussel reproduction and fish use again, and kind of restore the watershed. So there's a lot of cool little system sentinels in there. Also, they're great food. Sturgeon love floaters. Sturgeon eat quite a few of these. Historically, a lot of the native tribes use these for food sources, especially east of the mountain. A lot of raccoons, otters, lots of animals love nibbling on these mussels. They're reliable. They're always in the same spot. They're full of calories. I don't think they taste very good, but they are reliable and always there. This is another quick demonstration of how they're important to water quality and water health in the river. So this is one of my thesis spots. This is right below the South Elmo Bridge, and we had, we counted like around 100,000 pearl shell mussels there at the time, and ridge mussels, and it was really great. And so we were out there. We set up these two little aquariums, and did this fun little time lapse experiment. And this is something that's kind of fun to show to kids as well as adults. But here's kind of my still frame, like 30 minutes into it, you're not seeing a lot of change. And sorry, I ruined the experiment. One of these tanks has mussels, and the other one does not. So this is kind of through time. At like an hour and 45 minutes, without touching them, you can start to see the mussels in the tank on your left. And then in a little over 2 hours, you can actually see the gravel and the grass through the tank. And then in about 3 hours, you can just kind of see the tank with mussels in it, totally clear. And the one without, you can still see the turbid water. So these are just a couple mussels in a couple hours cleaning out a 5-gallon aquarium. And these mussels are filtering water 24 hours a day. And think about just hundreds of thousands of mussels at the bottom of the river, what they can do for water quality in the river. So we're kind of going from the cool part of the talk where we talk about how awesome mussels are to some of the issues that are in front of the freshwater mussels today. So I'll try and speed this up. I know I've been talking 40 minutes already somehow. So freshwater mussels as a family are the most imperiled group of organisms in the West, or in North America. About 70% of them are imperiled or extinct already. These are old reports now. It's hard for me to believe, but right here, this little orange part says like about 25% of the mussels are actually in a stable condition. This is comparing the family of freshwater mussels to other organisms out there, and just showing that 69%, this exactly what I just said, like 70% of them are in some sort of like bad state or declining state or extinct in North America. So here, the bad news part. I thought I fixed this, but anyway, these are some of the challenges for freshwater mussels that are starting to decline pretty rapidly in the Northwest. So of course, urbanization, our dams, obviously those aren't new, but they're continuing to cause issues for a number of freshwater species. Host fish, declining salmon populations, you can imagine, impact the ability of freshwater or of pearl shells to reproduce, along with competition with other species. So keeping that host fish population healthy is important. I meant to strike this one. Oops, sorry. I meant to strike out restoration projects. And that's really one of the great successes of a lot of people that aren't myself in the last 10 years or so, of really bringing best management practices that did not exist for freshwater mussels into the limelight. So the Xerces Society developed a whole workbook of how to work in freshwater environments with mussels. So a long time ago, or 10 years ago, we were really focused on salmon restoration and overlooking the mussel aspect. And that's really changed in the last 10 years. A lot of the older fish passage and rechannelization projects kind of left out mussels, despite being amazing projects. And now we're really seeing that aspect of both fish, freshwater mussels, and other freshwater native species being incorporated into those best management practices. Water quality is an issue for these species that can quickly become overwhelmed by poor water quality. Loss of habitat, invasive species moving in. Of course, we had the zebra mussel scare not too long ago, which hopefully was just a scare. And I forgot to add disease, and that's one of the new and frightening things happening out there. So I wanted to take you back to my thesis spot where I talked about having 100,000 mussels just out there filtering the river and cleaning up the river for us. In the past couple of years, that bed is pretty much vanished. There are still a couple mussels there, but that is essentially gone at this point. And we're seeing those declines across not just the Chellis River, but across other rivers in the West. And so we've had this really rapid loss of freshwater mussel abundance in a lot of our large rivers. So those mussels are mostly not there anymore. And that was, one of my thesis spots, a place where I took schools to go show them how amazing and abundant freshwater mussels were, kind of a showcase spot. And it's vanished in the course of 10 years or so. I'm going to skip right over this slide and go to the next one. So this is a bit more of a good news. So when I was starting off, this is all the information we had about freshwater mussels in the Chellis. These are the different little symbols there, the different types of mussels. That has greatly increased in the last 10 years. So we really have a much better picture. Alexa found another dot that's not on this map. I didn't have the foresight to make the map that shows all the new locations, but people, just like people in the Grays Harbor Stream Team and other interested parties, are just adding dots to the map. So we're really getting a better picture of where they are and what habitat they're using in the Chellis. So thanks to all of you who have reported freshwater mussels. That little heart-shaped, sorry, I forgot what all those were about. Quickly, I want to tell you about all the new stuff that's going on in the Chellis, like the past 5 years. So we just mentioned those surveys. So we're getting a lot of survey. We're actually getting funding to do surveys for mussels. A lot of that work's been led by Washington Department of Fish and Wildlife, U.S. Fish and Wildlife Society, the Xerces Society, and the Chellis Tribe. So we're all up there. There'll be weird people in dry suits snorkeling around looking for mussels this summer. This is actually a really well-timed talk, Alexa, because it's getting warm. The water is starting to go down in Chellis, and in a month or two, it'll be prime time to start looking for freshwater mussels in the basin. So nice job there. We're using new tools to locate them. So the state's been using environmental DNA, which is just a way to filter scrap DNA out of the water and then run tests to see if the specific DNA sequences of freshwater mussels are present in the watershed. So that really helps us like hone in on where they might be, which tributaries, which reaches of the river. We're trying to figure out what healthy mussels are right now. So there's a lot of people doing studies to understand what's a healthy mussel, what's not a healthy mussel, kind of enabling us to understand the health of individual mussels and their populations, because we were purely counting mussels before. Now we're starting to develop metrics that'll teach us if a mussel is healthy or its health is declining, or how to kind of assess those mussels. There's been a really tragic disease. We believe it's a disease that's been knocking out mussel beds in the Chellis and other rivers in the West and the East. The Chellis River itself is kind of the focal point for understanding that disease. So we have people out drawing blood for mussels, just like you probably got a blood draw. At some point in your life, we're drawing blood for mussels and we're trying to identify a pathogen and understand exactly what is happening. If it's a virus, a bacteria, something environmental. But it's been really amazing to watch and sad to watch this. We've been trying to find a reference site in the Chellis that's our healthy bed compared to the not healthy beds. And we can't keep up. We keep finding farther upriver, it's like, okay, this will be our healthy reference control site where things are going well. We'll find it one year and then we go back to sample the next year and that bed is halfway gone. So it's really been scary watching this disease. So we don't know exactly what's going on. We just know it's bad. And we're trying to figure out what's happening before we lose a lot of the large beds in Chellis. I would say the West is really becoming a lot more engaged in the national freshwater conservation work. We're our representatives from the West are going back east, talking to the people that have been working with freshwater mussels for decades. We're really starting to be engaged a lot more. People are asking us questions now, and are really excited about the work we're doing at West. So there's a lot of interest in Western mussels all of a sudden. And also, part of the reason we're here today is because people are really starting to recognize and learning to love mussels out there because of all the things they do for the rivers and all they're indicative of. So yeah, it's really exciting to have people to talk to about this. So really, this is part of talk where I can tell you how you can help. We're still trying to map their distribution, not just in the Chehalis, but all the watersheds. The ridged mussel is only known from the Chehalis and 2 rivers in Oregon west of the Cascade. I'm sorry, the ridge mussel is only known from the Chehalis and 2 other rivers west of the Cascades. So it's really unique to have that species here in Western Washington, but I can't believe that's the only river in Washington west of the Cascades where the ridge mussel is. So I'd really, really like to, know more about ridge mussel distribution, but all mussel distributions. Citizen science groups to monitor kind of the population health, to let us know if other populations are blinking out. Sending information to the conservation agencies. Right before we started, Alexa was showing me her image behind her, and that's sort of like, hey, I found dying mussels on the side of the river last week. That sort of information is really helpful to us. And this last bullet, again, look before you restore and dewater. That's kind of gone off the list now because we know about freshwater mussels now, and we have the ability to talk about them and know what to do when we do encounter them now. Kind of the same slide. We have an iNaturalist site, and I'll send all these resources to Alexa after this meeting. But just posting a picture of kind of skeptical kids holding up a mussel, that's information, that's data for scientists to know. Distribution, where we can serve, new study sites, new questions, it's all great. It's a picture of mussel monitoring, pretty fun stuff. Teaching the next generation about these mussels. This is kind of, a lost bit of information that we're starting to bring back into the conservation community and conversation right now. So getting people to know about freshwater mussels is great. And sorry, I was trying to rush through this last couple ones, but didn't realize that I got talking so loud. So this is a test. Alexa might be able to pass it because I showed this last time. But while you're pondering any questions you might have for me, I challenge you to find the freshwater mussel. There's one freshwater mussel in this picture. This is one of the reasons they're hard to find sometimes. I'll give you 2 hints. It's a pearl shell, and it's about in the center of the screen. I know you can't show me, but I'll highlight it in a second or two after you have a chance to ask any questions, or Alexa tells me it's time to get off the stage.
I'm not seeing any questions in the chat, but if people have questions, please feel free to speak up.
So this is Martin. I do have a question for you, Teal. With fish, the water temperature in the streams is critical. I mean, it can lead to mortality if there's a water temperature change. How does it affect freshwater mussels? Is because during the aquatic species restoration plan events, it showed how there wouldn't be much remaining cold water refugia in the whole of the Chellis Basin over time as we encounter climate change.
Yeah, certainly freshwater mussels are very sensitive to that as well. It's a little species dependent too, as you can imagine. The floaters, the anodonta, they're used to the lakes and shallower, kind of muddier habitat, so they can tolerate a little warmer water. But the pearl shells, yeah, that's one of the questions we're pursuing, especially with these large bed depths in the Chehalis and other rivers, is maybe it's not a new thing that's in the environment. Maybe they're becoming temperature stressed and just they're more susceptible to bacterial and virus loads than before they were in better shape. So I believe there is work. I believe I believe it's publicly available, that kind of is starting to look at the same kind of like when the temperature gets too hot for specific salmon species. The United States Geological Survey is also working on information for that kind of in the West for pearl shells as well. They've decided to use that as kind of an indicator as well. So that's, I think I can dig that up. It's a little harder to find than the fish stuff, but that temperature sensitivity is certainly there for these species as well. Has anybody found the mussel yet?
Laura
Looks like
said she found some. Sorry, Kurt.
I was just saying, it just looks like off to the right of the clipboard, about an inch or two. Right, just off center.
Yep. Am I getting close?
To the right, to the right, to the right, right there.
Oh man, you found it. Yeah, that's it right there. Nice job.
It's a little bit
That's-
darker than the rest of the stones.
Yeah, so that's actually in Delzine Creek, kind of coming in between Porter and Elma, I think. So that's up near the Boy Scout camp up there.
So Teal, I have a question. It seems like all the photos, they show larger Western pearl shells. Is that just because we like to shoot old trees, old growth trees? Because there seems to be some concerns in Oregon about there's older shells, but they can't find any of the younger age class. Or is
Yeah,
it
that's
just-
certainly. Oh, sorry. Go ahead.
No, I'm just kind of curious. Do you find that in Washington that we see, or we just like to shoot pictures of just big clams? I mean, big
Yeah.
mussels.
Yeah, that's definitely true. The old growth photos are the best and the most photogenic. But yeah, juveniles, they're further in the substrate a lot of the time, so they're not quite as apparent. But certainly, what you're saying is true. We do see a lot of lack of juveniles in some of these populations, and sometimes that's realized in like all of a sudden all the mussels are gone and we just realize there's been no reproduction for the past 50 years or something like that. And you know, these big shells like we see at the bottom of the Chehalis are making everybody seasick now. You know, we see this for 50 years, you know, it's mussels, and then eventually they're old and pass on, and there's nothing underneath them. There's no one there to take their place. So that's certainly true. We do see juvenile mussels fairly frequently in the Chehalis River. So we don't think it's a recruitment issue, but in many cases it is a recruitment issue. That's great observation.
The other question I had was the map of the Chellis Basin that you have. Did you do any work above the proposed dam site? Above the town of Teal?
Yeah, certainly. So okay, this one's good enough. I think that's right. Somewhere around here. Is that right? That's PL. Or is it further? But
further.
yes, I guess this is maybe the other map's better. We did do, there are a lot of surveys, certainly as part of that large aquatic ASRP, aquatic restoration species restoration plan, aquatic species restoration plan that's associated with that. Sorry, to get my acronym straight. They are conducting surveys throughout the basins, including the Olympic drainages and a lot of these cascades and Willapa drainages. So I think they've been, I can't remember if it's the South Fork or the West Fork that we haven't found freshwater mussels in yet. But yes, there are kind of basin-wide surveys in the Chehalis right now. I think some of that information should be up soon on that Chellis Basin ASRP. I know some of those reports are coming due. I'm not a part of that effort, but I've been kind of following that work. Yeah, and it is something it's very easy to contribute to. Last year we were riding that great Willapa Hills Trail with my family and stopped off for a swim and found a bed of mussels that I couldn't find any record of just when we were throwing the kids around in the water. So it's you know, that information is really valuable. And also, you know, we, especially with the pearl shells, you can be pretty sure where there's pearl shells, there's salmon use as well, or at least trout use. So that's, you know, a great indicator as well if you're looking to say this is important for fish and you find mussels, then you can say, yeah, it's important for fish. See, there's mussels here. Mussels can't exist without fish. And mussels are a lot easier to catch than fish in my experience, but I could just be a poor fisherman too.
So this bed behind me, a lot of them had died, but and that may have been due to the low flows, or this last couple of weeks. So I took this picture for everybody's reference. I took this picture on Saturday, this last Saturday. So just after those few dry days that we had. And there's quite a few dead here, but the ones that were underwater were still alive. So do you think that is like they just dried out during that dry period that we had? This could also be a section of stream that it's tidal in this location too. There's no salt water, but the Chehalis gets backed up here. So there is tidal influence. It's hard to say what.
Yeah, it's hard to know what caused that, but definitely that looks, I can't tell if there's any tissue left in the shells, but it looks like a fairly new event. They still kind of look oriented in a way that makes me think they were upright and alive not too long ago, or they got recently washed out of their bed in a pulse. I guess I don't know if this site is underwater, but that certainly is the signs that we find alarming and kind of like, oh man, what just happened? Because it's not a shell here or there, and the natural kind of die off you'd expect, but that looks like hundreds of mussels perhaps that died at one event. That's what we're most concerned about right now.
Yeah, this was near the Wakefield Bridge, near.
Oh yeah. So I know people are probably ready to get on with their evenings. I did want to note that we were petitioned last year for the Western Ridge mussel. The U.S. Fish and Wildlife was petitioned to look at the Western Ridge mussel under the Endangered Species Act. So we'll be hearing more about that soon, I'm sure, but something to have on our radars. I'm trying to quickly post in a couple resources into the chat box here, but Alexa, like that, we do have, there are places you can report those die-off events too. And I'm trying to deal with my little screens here though. So that sort of information is really, really cool and valuable.
Yeah, I'll definitely report it.
A mention of a new bed in the Wishkah too, which is great.
Yeah, Laura said she found a bed in the Wishkah, and she posted the GPS points that she found it, so I can send those to you, or you can copy and paste them out of the chat. So that's really exciting. So thank you, Laura.
Yeah, thank you. I won't recognize your face, but I'll remember your name and think of it fondly.
Well, thank you all so very much for joining this evening. And I have recorded this full presentation. So if you would like that, I can send it on to you if you put your email in the chat. Otherwise, I will probably be sharing it with the MRC, so it can be shared and used as long as that is okay with everyone here. But again, thank you so very much, Teal, for joining us this evening. You gave a really informative talk, and I very much enjoyed it and even learned some more things. So thanks again, and thanks for everyone for joining. And it was really good to have you here, Kurt and Martin and Laura. Thank you all so much for being here. It's good to hear your voices and have you guys around. And I'm so glad you could join this evening. So thanks again.
Thank you.
Everyone have a nice evening and have a nice rest of your week and weekend.
Thank you.
Thanks so much.
Thank you.
Happy mussel finding.
Yeah, and reporting. Finding
Yes.
and reporting.
Yes. Don't forget the reporting portion. That is very, very important.
Thank you, everyone.
Mhm.
Yeah, and
Bye.
please do reach out if you have any other questions.
Yes, for sure. Bye. Thanks again.